Hello, I'm Bill Hearl, CEO and founder of Immunomic Therapeutics. I really appreciate the opportunity to speak to you today, and as a attendee of the World Vaccine Congress for the last 15 years, it's been a real pleasure to be able to participate in this fantastic event. So thank you for the invitation and the opportunity to speak. Today I'm going to be talking about our GBM program for treatment of newly diagnosed glioblastoma using our targeted vaccine for cancer. So just a few words about Immunomic Therapeutics. We were founded in 2006 based around the LAMP technology that was developed in the laboratory of Dr. Tom August. We worked very hard with Dr. August to adapt the technology for commercial purposes, and then developed an initial indication in allergy, which is now actively in development by Astellas Pharma. As we were working along with Dr. August on a variety of topics, it came to our attention that Dr. Gilboa at Duke had done some very interesting work on cancer vaccines dating back to around 2005 and even earlier than that, using cell therapy. And so when the opportunity came to return to the immuno-oncology space when we completed our license deal with Astellas, we were able to immediately continue work on that platform, working with Dr. John Sampson and Dr. Duane Mitchell at Duke and the University of Florida, respectively. And over time, we developed this platform that we originally licensed from Johns Hopkins into what we now refer to as the Unite platform. And we have several major programs in oncology, all in the structure of a cancer vaccine, and essentially focusing on cancers that are related to viral antigens. So our lead program is a clinical stage cell therapy to treat newly diagnosed glioblastoma or GBM. The phase 1 data, as you'll see, had excellent long-term survival, and we have an active phase 2 study. Over 120 patients now, I'd say more like 135 patients are enrolled, and roughly 100 patients under treatment. So the key components of the Unite platform include our universal intracellular targeting expression using the LAMP platform to specifically target MHC2 and activation of helper T cells. We've augmented the original technology not only with new improvements to the LAMP design, but also with adjuvants to enhance immune response, a variety of delivery options, and large-scale manufacturing potential. So really all the components that you need to create an effective vaccine have been pulled together and united, if you will, into a single platform. And one of the key elements of this, as you'll see, is that it induces a strong helper T cell response while maintaining its cytotoxic T cell activity that you would expect with a nucleic acid vaccine. So you get a broad-based immune response. Historically, in terms of applying this platform in oncology, there's been multiple programs, and all of them have shown clinical benefit to the patients. So as I mentioned, we'll spend most of our time talking today about glioblastoma and the CMV targeted PP65 protein as the basis for our work with Dr. Sampson and Dr. Mitchell. We also have been working and still are collaborating on designs that incorporate human telomerase and also our cell therapy. And this work had been conducted specifically a fairly significant study in AML, where they had significant clinical benefit long-term in high-risk patients. So there's a second target and a second indication where the technology platform was successful in an oncology platform. And then also some work that was done in Brussels by Dr. Thielmans with melanoma, targeting a couple of these combined with his trimix technology, also was successful in inducing a clinical response. So I just want to take a couple minutes and tell you how the technology works as far as targeting, and go through that just very quickly. So LAMP is the abbreviation for lysosomal associated membrane protein. It is an essential protein that is affiliated with antigen presenting cells, specifically a highly glycosylated protein that is found in the membrane bound in the lysosomal membrane. These vesicles specifically contain the MHC2 molecule, so you have the ability to directly load the MHC2 molecule for presentation. And so it takes advantage of this natural intracellular localization sequence to bring the antigen to the key antigen processing center in antigen presenting cells. LAMP itself is a kind of a paperclip design, I would say, and it's got three major domains: a cytoplasmic domain that helps target the nascent protein as it's synthesized into the Golgi for localization into the lysosome, a transmembrane domain, and then a luminal domain that is multi-component connected by a hairpin. So we take advantage of this molecule to directly target the antigen into the lysosome. And basically what we imagine happens, and is based on a good deal of immunohistochemistry and confocal microscopy, you can show that the LAMP antigen fusion protein does indeed co-localize with MHC2, and then as the lysosome matures and the pH drops in the enzymes for digesting proteins become activated, the target antigen becomes cut into small peptides, and with the release of the clip protein is now free to bind to the MHC2 molecule. The end result is that MHC2 presentation occurs on the outside of these antigen presenting cells, the dendritic cells, and it is able to directly interact with naive helper T cells and educate those cells and result in a specific antigen presentation. So practically, on a little bit larger scale, how does this result in attacking tumor cells? So you actually have a bifurcated pathway where a certain amount of the antigen-linked protein still goes through the proteasome. So you get simultaneously MHC1 presentation and MHC2 presentation in these cells. And the CD4 cells, as they become activated, enhance the cytotoxic T cell activity. They also will migrate into the tumor along with the CD8 cells, and you end up with an enhanced killing effect. You also get a very potent antibody response as a result of this MHC2 presentation. There's, we believe, perhaps a endosomal type of pinching off at the membrane that seems to have a large amount of antigen associated with it that can directly interact with B cells. So in the glioblastoma arena, what was discovered about 20 years ago, really, now, is that by Cobbs and then confirmed later by Mitchell and et al., is that there's a high degree of correlation with presence of CMV in GBM tumor samples. And so in Cobbs, he found it in all his samples. Dr. Mitchell found it in 91% of his samples, as you can see, and there's a strong staining for PP65 in these. Now, it's important to note that CMV is not believed to be any sort of causative agent, but more likely a secondary infection in an immunocompromised individual. So you have a very unique target where you can take advantage of the fact that the CMV is present and use this to bring cytotoxic T cells into the tumor and affect the survival of tumor cells. So the process for autologous dendritic cell therapy, I'm sure is very familiar to many of you. Our partners in this, Aldevron has been manufacturing our messenger RNA, and we're working with Commune to go through the manufacturing process. So following isolation of a variety of white blood cells through the apheresis process, those using Commune's proprietary process use a variety of growth factors to create cells that can be loaded with the DNA or RNA in this case, and result in creating mature dendritic cells that specifically target the CMV-PP65 protein. And then these cells are returned to the patient in a subcutaneous injection, and they actually have been shown to migrate to the draining lymph nodes, and that is the process. It takes about 7 days or so to actually go through this manufacturing process, and while that's going on, the patient is undergoing standard of care. The result for this in the original ATTACK study is that the patients had long-term survival benefit. So in this first study, the number of patients having long-term survival was at about 50%. And we took two approaches with this. It was either with standard radiation and TMZ coupled with pre-treating the injection site with tetanus toxin to attract lymphocyte cells to the injection site, or alternatively using GM-CSF and getting similar data. So as you kind of take this collectively, and you can see that that's rather remarkable survival benefit in terms of, especially relative to historical controls, where you're looking at a normal survival past initial surgery of about 14 months and a 3-year survival percentage of a very small number, a few percent of glioblastoma patients live beyond 2 years. In these patients, the long-term survival was years. And so the patients that made it out to the situation where they were having a long-term benefit in either ATTACK 1 or ATTACK 2 lived for many years. And in fact, they did not die from glioblastoma. So if you look at this in a tabular form, you're looking at the overall survival at 36 months, 40 months, 60 months is really remarkable in patients that were treated with the cell therapy. What is unclear is with progression is how much of that may be due to pseudoprogression. There seems to be some trend on that, and I think we'll get more information on that with the ATTACK 2 study as we go forward. But you can see, and compared with the placebo comparator, which was standard of care, the 36-month survival was 16%, and the 48-month survival was 0. With the GM-CSF, you had very similar numbers, 36-month survival at 50%, 48 at 36, and 60-month also at 36%. So those are rather remarkable numbers for survival for the ATTACK study. So this is ATTACK 2, and this is ongoing. It's a 3-arm study, 2 arms of therapeutics and 1 placebo comparator. It's a blinded study. It started, enrolled its first patient in 2016. We anticipate completion of enrollment this year and being able to get to an endpoint readout shortly thereafter. The alternative to the cell therapy is a plasmid DNA design, and in this case we were able to look at the possibility of expanding the patient eligibility in this by going with a DNA vaccine, so there's no need to draw cells from the patients. And you have the ability to have this as an off-the-shelf treatment, so there's no waiting to begin treating patients. In fact, as you'll see, the study design will start treating patients before surgery and beginning radio before they get their radiation treatment or TMZ. So it really has a lot of options for the patient. And in this case, you can see the targeting both PP65 and ID1 or the GB protein induces strong responses in interferon and antibody in these subjects, in those mice studies. And in animal modeling, you end up with, again, significant survival in a very aggressive tumor model where the untreated mice die very quickly. So our phase 1 study design now is to give the patients 3 doses as they get through surgery and before they start their initial treatment with the TMZ, or if the patients are unmethylated, we're going to skip the TMZ maintenance, as that's been shown to have no clinical benefit. So we'll be able to do a little bit of a comparison of the impact of TMZ maintenance on our trial subjects. So we expect to get this started at the end of the summer. We're on the verge of filing this IND. Everything seems to be in place, so it's a matter of paperwork now. Clinical sites are in place, and we are ready to go. So we're really quite excited about this. And I'm going to stop there. I thank you very much for your time and listening to my presentation. Please feel free to present any questions in the chat box or email them to me, or you can post a question on Twitter. So thank you very much.